Thanks everyone for coming to our Keck Seminar today. And so this is a seminar series by the Gulf Coast Consortia here in the Texas Medical Center. And it is a trainee-led speaker series. So we're really excited to have our speaker today, who's Dr. Durre Mohammed. She's an assistant professor at the University of Massachusetts Amherst, and she was also a trainee here in the TMC as a postdoc in Bonnie Bartel's lab at Rice University. And she was she was also a K99 Mosaic Fellow in that capacity. And today she will talk about her current lab and her journey of how she got there, and her lab studies peroxisomal periostasis. And yeah, take it away, Dr. Mohammed.
Thank you, Alex, for the wonderful introduction, and thank you for inviting me to give a seminar for the Gulf Coast Keck Seminar series. I miss Houston right now, especially with the snow, the back-to-back snow we're getting here in Massachusetts, but it's lovely to join you all via Zoom. So today I'm going to talk about my journey in plant sciences, and also try to convince you all that plant science has a deep impact on human health in various ways. As Alex mentioned, I am an assistant professor here at University of Massachusetts in the Biochemistry and Molecular Biology department. And this is a new position. So when we get to the question section, please ask questions about my journey, about my research, but as well as some of the grants I received as in the job market and what it's like to start a lab. As again, I've started in September, so I'm pretty new to this. So my personal journey, I'm born and raised in Chicago, Illinois. I attended University of Illinois at Chicago, where I got my BS in biological sciences. While I was, I guess, a sophomore going into my junior year, I had my first daughter, seen here, Kenya, who really took this journey with me as well. So she's very important to me in that respects, seeing this journey go this far. While at University of Illinois, I mentioned I had my daughter, and that experience in undergrad wasn't the best for me. I am a first generation college student. I didn't have people assisting me with this process in biological sciences, and I didn't enjoy myself at University of Illinois when it came to that field. And so when I graduated, I joined a non-for-profit that focused on women in the C-suites and how to navigate that process. And so I went back to school to get my MBA because I was planning to leave science behind. But what I want to say is that even though I didn't enjoy myself at University of Illinois, there was a key person, Dr. Catherine Wapayer, who kind of took me under her wings towards the end of my college career, allowing me to come in her lab and do some research. And that helped me have a perspective where the glass was half full, not half empty. Her tutelage really, I guess, was important for me to come back to the world of science. She introduced me to Dr. Terry Long, seen here, someone who had shared experiences as I had. And between Dr. Terry Long, Catherine Ropea, and Danielle, a graduate student in Kate's lab, they really set the foundation to allow me to really grow and explore in this field. I left again to go work at this not-for-profit, but Catherine was not accepting that. Terry Long was just finishing up her postdoc at Duke University and was starting a lab at University of Illinois and was going to need a lab tech. And she heavily, Kate recommended that I take this position, even though she knew I wanted to kind of leave biology behind. And so I took the position, and I could say that that was one of the best decisions I ended up making. I ended up working for Terry Long, who took me under her wings, and really I blossomed in her lab. And so much so that she encouraged I go to graduate school because she ended up leaving UIC and went back to North Carolina, where she's from. And she encouraged me to go to graduate school. She let me know that graduate school, as a parent, was possible. And so I did. I eventually went to North Carolina State University, where I obtained my PhD in the plant and microbial biology department. I did okay there. I received a couple awards, such as the excellence in doctoral dissertation. So each department in the college put forth a nominee, and all of the nominees, I was the one that won. And more recently, even receiving the Outstanding Young Alumni Award, which just highlighted the progression in academia that I've been on. After leaving NC State, I went to Houston, where I joined Dr. Bonnie Bartel's lab at Rice University. I also got married and had 2 additional children. And I had a very fulfilled life at Rice and in Houston, here with a colleague of mine, Catherine. While there, I also did pretty well when it came to funding. I started off with a Rice Academy postdoctoral fellowship, followed by NSF postdoctoral fellowship in biology, and then NIH K99R00 postdoc fellowship. And that kind of assisted with my next position as assistant professor here at UMass. And so here's a picture of myself and my 2 youngest hiking, which is very beautiful, and a lot of hiking trails here in Amherst, Massachusetts, while my oldest daughter, who was along this journey with me the whole time, headed off to University of Maryland for her first year of school. Throughout this process, a common theme was that I worked on plants, but I worked in areas of plant research that heavily impact human health. So at University of Illinois, this work revolved around plant secondary metabolites, particularly the anti-UV properties that these secondary metabolites can exhibit. And then also we use this as in collaboration with people who work in cancer as a way to prevent cancer cells from growing. And so again, this direct connection to human health. And I wanted to stay in that path. And when I moved to North Carolina State University, I shifted from plant secondary metabolites to iron homeostasis, where we really focused on a transcription factor that induced a transcriptional cascade for the plant to be able to survive in iron limiting conditions. And I appreciated this because this specific transcription factor is not just in plants, but also in humans. And so recognizing that proteins found in plants have severe homology to what you will find in humans, and what we can identify in plants, the mechanisms behind these proteins in plants could be directly applied to humans, was also exciting to me. From there, moving to Rice University, I wanted to shift from just focusing at the transcriptional level to more at the protein level and even the organelle level for cell biology. I moved into peroxisome maintenance, where I focused on proteins that were essential for maintaining the homeostasis of peroxisomes, as well as processes involved in selective autophagy of peroxisomes, termed pexophagy. So what are peroxisomes? Peroxisomes were one of the last organelles identified. Oftentimes it's still misidentified and it's understudied. But Christian received a Nobel Prize in medicine in 1974 for his discovery, shortly after the discovery of lysosomes. And around the same time, there is a wonderful plant biologist called Harry Beavers, who also identified, at the time, he called them galaxosomes. And what we know now is that the galaxosomes and peroxisomes are the same organelles. And so just to let you know, we find these organelles in mammalian systems, fungus, and as well as plants. So all eukaryotes, they, as I mentioned, if you are aware of them, people commonly identify them as the organelle that performs beta oxidation or ROS metabolism, but that kind of cells this organelle short, and that's because they compartmentalize a diverse enzymatic reactions. And so mammalians, and the diversity spans across organism. So in mammalian systems, they also carry out alpha oxidation. They're the site for phospholipid biogenesis, which is essential in brain development and myelin sheath, as well as bile acid biosynthesis, having some role in antiviral immunity. And fungus, again, various metabolic pathways that impact growth and defense. One of the key discoveries in fungal peroxisomes was that penicillin, the biogenesis occurs in peroxisomes. And then in plants, their role in photosynthetic efficiency, herbivore defense, sex determination, growth and development varies with the different pathways that take place in peroxisomes as well. These different pathways don't act solely in peroxisomes. There are a lot of metabolite exchange between, say, the mitochondria and the chloroplast. There's collaboration with lipid droplets for the fatty acid beta oxidation. And there's also interaction with the nucleus for some retrograde signaling. So there's a lot of interorganelle interaction and collaboration between peroxisomes and the other cells. And so one might imagine that it's essential for peroxisomes to maintain its health. And they have various processes that contribute to this maintenance. They house reactive oxygen scavengers, they house chaperones and proteases. These work separately to maintain the overall health of the peroxisome membrane, as well as proteins that localize to the peroxisomes. Peroxisomes also have this ubiquitin system where proteins could be polyubiquitinated and taken off to the proteasome for degradation. And kind of a final line of defense is autophagy. So when peroxisomes are obsolete or damaged, there's an unknown signal that we are working to uncover that signals or targets it for degradation via autophagy, taken to the vacuole or lysosomes and mammalian systems for nutrient recycling. And so why is this important? There are congenital defects are presented at birth where there are causal mutations in, say, PEX1, PEX6, and PEX26. These are various peroxisomal proteins. And we know that these proteins suppress pexophagy. And so that lets us know that pexophagy is a major dysregulation of pexophagy is a major characteristic for peroxisome biogenesis disorders. And peroxisome biogenesis disorder presents in various ways shown here. We also know that the peroxisome or peroxisome proteins, the machinery for autophagy, and as well as LAN2, a protein that I heavily worked on, are conserved across from plants to mammalian systems or humans. And so the work we do in plants could inform the work being done in patients as well. Beyond these congenital defects, we also know that over time, dysfunctional peroxisomes could lead to other diseases or defects, such involved in neurodegenerative diseases, aging, viral infection and immunity, obesity, and diabetes, as well as cancer. And so the medical world is beginning to catch up on the importance of peroxisomes and peroxisomes maintaining their health. But in our lab, we focus on peroxisomes and Arabidopsis, and that's because Arabidopsis is a nice model organism to work on. In particular for peroxisomes, we have some very simple readouts. I mentioned that beta oxidation occurs in peroxisomes, but in mammalian systems, that could also occur in the mitochondria. And so in the plant system, this only occurs in peroxisomes, which allows us to have some readouts that are robust as well as very directed, knowing that these defects are specific to peroxisomes and not to the mitochondria. One of these readouts is an IBA responsiveness readout, where we add exogenous IBA, a precursor phytohormone that is processed in the peroxisomes. And if we add this exogenously, the plant roots will begin to change its root structure and produce more lateral roots. If this pathway is dysfunctional, what we know is that that doesn't occur. And so this is a good readout for determining whether or not peroxisomes are functioning well. Another readout that we like to use is this luminal import assay, where some proteins that are imported into the peroxisomes have their peroxisome targeting signal cleave, shown here. And this cleavage actually is distinct enough that we could see this on our amino block, where here I'm showing you an example of 3 various mutants that have a spectrum of severity of import. This first mutant, we are seeing no import, whereas the second mutant and third mutant, we see some import, but there are some defects going on with it as well. Another reason why we work in Arabidopsis is because the peroxisomes are larger in plants, right? So plant cells are larger than animal or yeast cells, and by extension, that's the case for peroxisomes as well. So sometimes we could get as much as 10 times the size of peroxisomes in plant cells compared to other model species. And for this reason, it makes it a nice model organism to work with when we want to do live cell imaging and look at the dynamics of peroxisomes compared to other systems. And here I'm showing you a difluorescent reporter line where we have the peroxisome membrane in green and the lumen false colored magenta. And so in this image, I'm showing you that peroxisomes, what were previously thought to be just a single membrane with enzymes stuffed inside, we recognize that that isn't the case, and that they're a little bit more dynamic, similar to chloroplasts and mitochondria, where they have structures inside here called interluminal vesicles that are formed via the escort machinery. And so this kind of visualization or realization wouldn't have occurred if we were working in cell cultures or cells that were smaller. So my lab's broad question is, how do peroxisomes maintain proteostasis and by extension whole cell homeostasis in high oxidative stress environments? And that's because, again, these metabolites have to be exchanged. So if peroxisomes aren't healthy and aren't able to export these metabolites, how is that affecting other organelles that rely on this transport? I mentioned that during my postdoc time, I focused on line 2, which I'll go into more details, but my lab here at UMass is interested in kind of all of the proteases that localize to the peroxisomes, as well as the chaperones and the oxidative scavengers like catalase and the superoxide dismutases that are in peroxisomes as well. But today's talk, I'm going to focus on line 2 and some of the preliminary work I performed at Rice University. So line 2 is a multifunctional protein, functions as a hexamer. It has an N-terminus domain here in gold, a AAA-TPase chaperone domain here in blue, and a C-terminal protease domain here in purple. When we mutate line 2, causing it to be dysfunctional, we know that we have fewer and larger peroxisomes in these plants. And this is a 3-day-old image, or this is an image of a 3-day-old plant where we have marked the peroxisomes with GFP. So compared to wild type, they're larger and we see fewer. We also know that the IBA pathway is dysfunctional. So here's a readout where we are measuring lateral roots or counting lateral roots per the main root length. And what we know in Nalan2 mutant, we don't see that increase in lateral root formation. So there's a defect in the phytohormone production. We also know that broadly speaking, there is a delay in germination and impaired growth compared to wild type. And finally, we know that there's a processing defect. So protein import into the peroxisome seem to be slightly less efficient or defective. Our lab performed a LAN2 suppressor screen where we took a LAN2 mutant and mutagenized it with a chemical called EMS that induces random mutations. And what we were looking for in this LAN2 mutant now was the recovery of lateral roots shown here in wild type. So upon mutagenization, we found some suppressors that did recover the lateral root formation. And then we rescreened that to see whether or not these suppressors also restored the peroxisome size. And we found that as well. Whole genome sequencing was done. And here I'm just showing you one of the suppressors, which is ATG2. We found, which is an autophagy gene involved in lipidation of the autophagosome, essentially expanding the autophagosome. And when we take a look at the double mutant, so aligned to at ATG2 double, we see recovery of most of the phenotypes. So recovery of the lateral root formation, recovery of import, and partial recovery of the overall size of and development of LAN2 defects. This was a very robust screen. We collected several suppressors, but all of the suppressors were mutants of the autophagy machinery. And so it was very glaring that it was time for us to take a step back and start to look at how a proteolytic system within the lumen of peroxisomes might be affected by a proteolytic cytosolic system, such as autophagy. So just to give a very simplified explanation of autophagy, we know that various cellular constituents, such as the proteasome, lipid droplets, mitochondria, chloroplasts, or aggregated proteins in the cell need to be discarded so that we can maintain the whole cell's health. And this process is done through autophagy, where a double membrane structure derived from the ER will target these different organelles. And I'm highlighting in purple ATG8 because this decorates the phagophore and is actually the docks with the signal on these various organelles. And I'm also highlighting ATG2 because that's one of the mutants we identified, and it's important for the lipid transfer to expand the phagophore and eventually close it. Once the cellular constituents, whether it be organelles or protein aggregates, are consumed by the autophagosome, it's transferred to or taken off to the vacuole or lysosomes, where they are broken down and nutrient recycling occurs. And so we went back to this initial phenotype of LAN2 where we see these large peroxisomes, and we wondered whether or not if the autophagy machinery are targeting all of LAN2 peroxisomes. And so to test this, we took a LAN2 mutant and we had a marker line that marked ATG8 with a GFP. Again, the ATG8 is the protein that decorates the autophagosome. And so here are our results from that. The top panel is going to be our GFP ATG8. Our middle panel is mRuby, false colored magenta, for our peroxisome targeting signal, so the lumen of peroxisomes. And then the bottom panel will be our merge. And so in our line 2 mutant, what we see is that the GFP ATG8 is co-localizing with not just the large peroxisomes in line 2, but also the smaller ones as well. And when we take a look at the autophagy double mutant, what we find is that we have this puncta-like GFP8 next to peroxisomes, but we don't see the surrounding occurring, essentially suggesting that the autophagy machinery is inhibited from completing its task of consuming the peroxisomes. So with these various phenotypes, the fewer and larger peroxisomes, we see recovery of line 2 phenotypes upon blocking autophagy, and then autophagy machinery co-localizing with peroxisomes. We came up with a model for line 2 function, where we believe that line 2 substrates are oxidatively damaged substrates that are damaged via the hydrogen reactive oxygen species that are accumulating in the peroxisomes, trigger pexophagy unless line 2 is there to disaggregate, degrade, or refold line 2 substrates. And so from here, we have a couple questions, which that we wanted to set out and answer. What are the broad wholesale consequences of line 2 induced peroxisome dysfunction? And what are line 2 substrates, particularly those that might be the selective autophagy receptors, in which mitigates the level of pexophagy occurring? To answer these questions, as a first pass, we took a broad approach where we use multiomics analysis to examine 3 different phenotypes. Our autophagy mutant, where we would survey the proteome and the transcriptome of blocked autophagy, including pexophagy. Our line 2 single mutant, where we'll survey the proteome and transcriptome for dysfunctional peroxisomes and dysfunctional line 2. And then our double mutant, where we're able to combine the defects, where we have dysfunctional peroxisomes, dysfunctional line 2, but we've blocked autophagy. And so taking a look at what that looks like. So here I'm first showing you our proteomics analysis, and this is a volcano plot where on our x-axis we have the fold change, and on our y-axis we have our p-value. And so I have highlighted in purple known peroxisomal proteins. And in our line 2 mutant compared to wild type, it seems that peroxisomal protein levels decline. We're seeing it out in our depleted proteome section. And it suggests to us that line 2 protects peroxisomal proteins from degradation in wild type. We went a step further and surveyed the depleted section via gene ontology analysis. And what we found was that terms associated with lipid catabolism were highly enriched in this population of depleted proteins in our line 2 mutant. And when we surveyed the proteins that accumulated in a line 2 mutant, we saw terms associated with photosynthesis and general stress response enriched in this in our line 2 compared to wild type. And then we moved on to analyze our autophagy mutant, again with this blocking autophagy. And we see a complete shift of where our peroxisomal proteins show up on a volcano plot. Now we see an accumulation of peroxisomal proteins in our autophagy mutant. And this suggests to us that peroxisomes are degraded via autophagy in wild type. When we perform gene ontology analysis on our accumulated proteins in our autophagy mutant, we see that we see an enrichment of lipid metabolism. So now we're seeing this complete shift in terms compared to line 2 mutant. And then we could do gene ontology analysis, enrichment for the depleted section. And again, in this section, this other metabolism are terms associated with photosynthesis. And so there seems to be this antagonist relationship between our LON2 mutant and our autophagy mutant. When we take a look at our double mutant compared to wild type, again, we see peroxisomal proteins in the accumulated fraction of our proteomics. And this suggests that LON2 protects peroxisomes from excessive pexophagy, and our gene ontology matches that of our autophagy single mutant, where we see lipid metabolism enriched. And so our double mutant is phenocopying our single autophagy mutant. And so we questioned whether or not this increase in autophagy was specific to peroxisomes, so selective autophagy of peroxisomes, also known as pexophagy, or if this increase in autophagy was nonspecific. And so here I am just highlighting again peroxisomal protein so we could see this very distinct shift from in our single mutants compared to our double mutants, where we have in our LON2 mutant depletion versus in our double mutant increase in these peroxisomal proteins. And we surveyed the proteome of other cellular organelles and cellular structures. One of the first ones we looked at was the vacuole. And we found that in our LON2 mutant, there seems to be a shift of vacuolar proteins in the accumulated section, which would suggest an increase in vacuolar activity when LON2 is dysfunctional, and kind of goes along with our theory that there's an increase in vacuolar activity because there's an increase in obviously peroxisomes being delivered to the vacuole to be degraded. And then we took a look at the other organelles or structures, and we didn't see this distinction like we see here with our peroxisome proteome or our vacuole proteome. We saw disturbances, but they weren't askew to either accumulation or depletion. But when we took a look at our plastid a little closer, we did see a slight skew in our LON2 mutant that was opposite of what we see with the peroxisome proteome. So it seems as if chloroplast proteome accumulates in a LON2 mutant. And we have some theories behind that that we're chasing down as it relates to carbon flux. So our takeaway from this analysis was that LON2 mutants induce autophagy is specific to peroxisomes. So we was comfortable with saying that in the LON2 mutant, we have an increase in pexophagy. So in summary, we found out that LON2 suppresses pexophagy, and we know that LON2 dysfunction leads to whole cell disruptions, which stood out to us was lipid catabolism and photosynthesis. So essentially, we're interested in chasing down the interaction between lipid droplets and chloroplasts in the near future. We next wanted to figure out how we can use these large data sets to kind of identify potential LON2 substrates. And for this, we kind of took an in silico approach where we removed the effects of autophagy. So essentially, we took our double mutant and instead of comparing it to a wild type, we compared it to the proteome that we found in our autophagy 2 mutant. And this is with the idea that if we have dysfunctional LON2, we get this increase in pexophagy. And so we need to block the pexophagy so that we could actually see what of LON substrates are accumulating and survey what that might look like. And so the hypothesis is that increased pexophagy is masking peroxisome and LON2 dysfunction, and we needed a way to separate that out. We also wanted to take into account if we're looking for an accumulation in proteins, that this could occur at the transcript level. where transcript is accumulating and then we're getting more protein accumulate. And so for this reason, we also surveyed the transcriptome and compared the proteome and transcriptome. So here I'm showing you a XY plot where on the Y-axis, I have our mRNA fold change. And on our, on the Y-axis is the mRNA fold change. And then on our X-axis is our protein fold change. In blue, we have transcripts that were significantly altered or differentially expressed. In gold, we have proteins that were differentially accumulated and that were significant. And in green, this was a matching of the transcript and protein that were both significantly changed. And so one might imagine that in this comparison where we have our double mutant compared to our autophagy single mutant, we're looking for protease substrates, and so they should accumulate. And so here we have these 17 proteins that are accumulating, and then we have these 285 that are accumulating, but the transcript hadn't changed. So here we have transcript that are decreased in protein accumulating and transcript unchanged and protein accumulate. We could take this a step further and we could analyze these proteins to determine whether or not they have a transmembrane domain. And this is important because if this is the selective autophagy receptor for peroxisomes, one would imagine that this specific substrate has to have access to LON2, which is an aluminum peroxisome, as well as access to the autophagy machinery outside of peroxisomes. So we looked for proteins that will span across the peroxisome membrane. And we also are interested in proteins that have AIM sites, which are autophage ATGA interacting motifs and would interact with ATGA on a phagophore. We could zoom in a little closer and just now take a look at how this looks for known proteins or known peroxisomal proteins, which I'm showing here. And this was important because this multiomics approach, we needed to verify that these results kind of match what we could find in our lab. And so in our lab, we had antibodies against some of these proteins where we were identifying. And so we tested whether or not this omics, this in silico approach really matched what we could do in the lab. And so I'm going to start over here. And what I want you to focus on is the column for the ATG2 and then the column for the double mutant, because this is essentially the comparison that we're looking at. And so ICL should accumulate in our double mutant compared to our single mutant. And we're seeing that we also took a look at PEX11 here, even though it doesn't look like it's significantly increased, there is an increase between of our double mutant compared to our single mutant. And PEX11 is especially exciting because this is a peroxisome membrane protein. We also surveyed MLS, which we see an increase, real significant increase in our double mutant compared to our single mutant. But then we also took a look at PMDH, which, if you notice here, suggests that only the transcript was significantly changed and not the protein. And our data shows that that looks to be the case. We weren't so lucky when we tried to find evidence to support proteins that were depleted in our data analysis. So here I'm showing you CSY3, which falls on this side here, right? So right here on the axis. And we weren't as lucky with our Western analysis of CSY3 in our comparison. And so we may have to find other ways to determine whether or not proteins that are depleted are really depleted, that we are finding depleted in our proteomics analysis is really depleted. So in summary, we were able to identify, using a multi-omics approach, we was able to identify potential LON2 substrates, some notable ones. when we focus in on proteins that localize to the peroxisome are obviously ROS scavengers, proteins involved or enzymes involved in fatty acid beta oxidation, and various other things. But we are not limiting ourselves to these proteins that we know localize to the peroxisomes, and we're excited about really identifying novel peroxisomal proteins as well. And so our next steps is to take a kind of a structural approach and utilize AlphaFold 2. We're getting help from a colleague here at UMass. So one of the nice things about transitioning to a new lab or a new university, a department, and finding your department fit, UMass has several different themes in their biochemistry and molecular biology department, one of which is protein homeostasis. We have plant biology, we have structural biology, and then neurobiology. And so we have core facilities and researchers who are really excited about structural biology and that will collaborate with you and assist with some of your work. And so our next step is to take our candidate line 2 proteolytic substrates, and we assess for transmembrane domains, but we're assessing for interaction with line 2 and using ColorFold, which is a script that was published in 2022. And Peter Chan here in my department kind of modified it, where we're not just looking at protein structure, but we're looking at the potential for protein interaction, where we have a bait protein, which is going to be line 2, and it goes through and predicts its structure, and then the structure of various pairwise Arabidopsis genes. And so we began doing that. We have a high computing system here that could do this in about 14 days when you're dealing with comparing about 10,000 genes, but the Arabidopsis genomes is much larger than that. And so we decided to take a step back and only put in our 330 candidate substrates and do a pairwise comparison of whether or not these are actually predicting interactions, as well as using these 330 with a similar platform to determine whether or not they have AIM sites. So really trying to get at and narrowing down our candidate substrates. And then again, we will obviously test and validate this. And so I have a graduate student right now who is performing a substrate trap and pull down and mass spec analysis. Essentially, we have mutated 2 different residues, one which will prevent the substrate from moving over, translocating over to the proteolytic domain, and then also killing the proteolytic domain. So we have this with a HA, so we could pull down line 2 via HA resin beads, and hopefully should get all substrates that are bound to line 2 because we are trying to inhibit it from being degraded. So these are our next steps, and we're excited about that. And just a point of clarity, this is done in our double mutant because, again, we want to make sure that we are not dealing with overzealous autophagy machinery, which will limit the amount of protein accumulation we should be getting when line 2 is dysfunctional. We also, again, we told you that we were interested in kind of the global effects or mechanisms when line 2 is dysfunctional because of the exchange of metabolites or collaboration with other organelles. And so from here, I mentioned, I showed you data that showed chloroplast, proteome, this that was heavily influenced and aligned to mutant, and also all of these terms associated with lipid catabolism. So lipid droplets are important for us to take a look at. And so I have one graduate student who's chasing down this lipid droplet peroxisome interaction question, and we'll have somebody else work with the question regarding dysfunctional peroxisomes and chloroplasts. All right. With that, I want to thank several people. And so I'll start back forward. I want to thank my graduate mentor, Dr. Terry Long, who really fostered my development and made a lab suitable for a single parent in grad school. I want to thank Bonnie Bartel, who she was an amazing mentor, and she and I have lifelong friends here. I'm highlighting um in her lab that to this day we still call each other and catch up with each other. I want to thank my collaborators because without them, I wouldn't be able to process all of the multiomics analysis. So Dr. Viestra at WashU, who performed his lab performed our proteomics analysis. And then my dear friend from graduate school, who's now at the Broad Institute, who process or analyze all of the multiomics analysis. I have to, of course, thank my funding sources, Rice Academy Fellows, NSF, and NIH. And lastly, I want to thank and acknowledge members of my new lab. So Patrick, he's a graduate student in our molecular and cellular biology department, biochemistry and molecular cell program, and he is chasing down the lipid and peroxisome story. We've performed lipidomics analysis, and he's been going through that data, as well as doing some lipid staining and thin layer chromatography to identify tag species and the decay of tag species. And also want to thank Eileen. Eileen is another graduate student, first year graduate student, who is focusing in on our substrate trap study and also running the pipeline to identify predicted substrates of Lyme 2 and also the AIM sites in our candidate genes. And then my lab manager, Haiping, who just started today, but she has a wealth of knowledge because she's been at UMass for over 12 years working in various people's labs. So she's been wonderful so far. And with that, I could take questions. And I'm going to put the screen up so I could see people. Okay.
All right, thank you, Dr. Mohammed. And if people can just unmute themselves and ask a question or enter a question in the chat, and afterwards I can just read that question if you have something to yeah, write in the chat box.
And this could be questions about the science. This could be questions about my path in science. And this could be questions about transitioning to your independent lab. I'm happy to answer all questions.
I'll start with a question. Um, so I guess my question is more about career development, as you just talked about, especially with the transition to independent career. I want to ask you, like, if you had like which kind of K award programs you considered for your independent career. I know you ultimately received the K99, but I was wondering if you considered the K22 or the K01, and kind of like what kind of decision making went into that.
Yeah. So when I asked Bonnie to join her lab, I told her that I would um, apply for grants to try to support myself. And so while in Bonnie's lab, prior to joining, I applied for the Rice University fellowship that I received. But once I got there, I applied for NSF postdoctoral fellowship in biology. I applied for a HHMI, the Hannah Gray fellowship, and then I also applied for the K99. I will say that I received the NSF one, and so that got me through a couple years. And I didn't get the HHMI. And I'm someone who kind of take universal signs to kind of determine my next step in life. And when I didn't get the HHMI, I don't know if I questioned, but I took it as a sign what maybe academia, the next step in academia isn't necessarily for me. But I didn't give up on it yet. And I think at that time they had just introduced this K99 mosaic. So it's under the K99, but it's mosaic, which essentially are looking for diverse perspectives in science. And so when I saw that, I said, okay, maybe this is time to try it again. And I applied, and I thought that I missed the first, like a first round. And I did a second round knowing that, okay, I'm going to have to apply again. And I didn't want to miss out on that span where I was going to be a postdoc for too long and didn't qualify. And I got it. So to your question, Alex, I didn't look into any other training postdoc fellowships from NIH at least.
Thank you. That was helpful. There's a question in the chat box.
Which
What do
I
you
can ask directly, because
Oh
maybe
yeah,
I
sure.
have the
Yeah, yeah,
wrong
absolutely.
slides.
Yeah, yeah, yeah,
Yeah,
yeah.
this is Catherine Shine from the UTMB. Can you go back to your slide on the selection of proteins that are binding to LON2, I believe? Yeah, just back. But anyway, the question was actually, are you doing some kind of control pull down there that the Google Doc is going to help you?
Hold on, let me get out of this, my screen. Okay. Here.
Yeah. Okay, everybody's
Yes.
trying to use Google Doc now for just this, but most of it's computational. Are you able to somehow select the proteins that you expect in the pull down?
So are you asking about the AlphaFold portion, or are you asking about the actual
I'm
substrate?
asking about the actual experiment here.
Yeah, so right
Do you
here I'm on
see
the
Google Doc helping you here?
is who helping me?
Is it, well, what he's doing is basically Google Doc. So it's a new one on AlphaFold, but I think it was originally called Google Doc, which means that you can look for proteins that dock onto
Yes.
your structure. And people are very excited about this, but it's computational at the
Right.
time.
So
So how do you see it's helping you?
that's separate from my actual experiment. So I have
Okay.
these a little over 300 candidate genes that I identified from my multiomics analysis that I'm running through that platform. Separate from that, I designed constructs that contain a HA tag, but also have mutations and various domains that that presumably will allow line 2 substrates to bind and stick for me to be able to do a pull down in a mass spec analysis.
Mhm.
And I
So,
have
so
various,
they're not really, they're just separate and you'll just
they're
do the
separate.
results.
They're separate, yes.
Okay, it'll be interesting to see how well they predict what you come out with.
Absolutely. I'm right with you.
Yeah. Also, you'll know where they're binding on your protein. So that's
Yes.
because
That will,
they'll tell you, they'll predict something for the interaction.
I'm sorry, I'm gonna,
It will predict where the interaction is occurring, and it'll give you the basis for more experiments.
yes, ma'am. I definitely agree. I just met with a student who's coming in next year.
Mhm.
And he's extremely excited. He has some structural background. And so I may get a student who actually began to mutate some of these binding residues and see how that looks.
Well, I wish you good luck, Durre.
Thank you. I'm sorry, I'm having difficulties where I could see who's talking and or not.
Ellen, do you want to unmute yourself and ask your question, or I can just read it too, whichever you prefer. Okay, I can just ask it. So the next question is, regarding your research, I understand you're using a plant model to study a cancer-related topic. How is it having to explain the strength of your model to the field of cancer, who mostly uses cell culture mouse models? And do you consider it a difficult portion of justifying your work?
Yeah, so this isn't necessarily for cancer research. I don't want to be a plant apologist. This is basic research in Arabidopsis that our findings can just inform people who work on quality control mechanisms for peroxisomes. And if that happens to be people who work in cancer, that's wonderful. And so I will say that peroxisome research in plants is far and few between. So most of the interaction that we have when it pertains to peroxisomes is actually in people who work in mammalian systems or who are clinicians working with samples from patients. How difficult is it to explain the importance of it? I don't. I think if you appreciate science, then it's not a difficult thing to appreciate to understand that overlap essentially.
question for you, if it's all right.
Hi, Gabby.
So regarding your collab fold, so obviously it has a big advantage of being able to screen.
You went out.
All right, can you hear me now?
Yes.
Okay, sorry about that. AirPod problems. So regarding the collab fold, so it obviously has a big advantage of being able to screen so many proteins through the computational difficulty of that. But is there an additional parameters that is testing how the interaction occurs, right? Because now AlphaFold 3, you're able to put in like multiple proteins and do complexes, but is there like additional parameters that CollabFold has that are to sort of understand whether this is like a real interaction or not?
It's not as advanced as AlphaFold 3, but it's doable. Like it's going to give us a metric of how strong it is.
Mhm.
The downside with AlphaFold 3 right now is that there's some licensing issues going on. And so we have not moved on to that. And I think for the purpose of me just kind of screening and seeing what that interaction looked like, knowing that I also have an actual experiment that would give me direct interactions, I think is sufficient for what I'm going to do right now.
Yeah, that makes sense.
Any other questions from the group? Any trainees? All right. If there are no other questions, let's thank our speaker. And thank you all for coming.
Thank you.